Welcome to the second part of our video series on gluconeogenesis. So in the first video, we discussed the details of the enzymes and substrates involved in gluconeogenesis. And in this video, we're going to recognize that when one pathway is activated, the other is deactivated. So in some cases, these are controlled by the exact same effector. So we're looking at the reciprocal regulation of glycolysis in gluconeogenesis. So if you remember, this is a small piece of our human metabolism puzzle here, right? And so for glycolysis, we were starting with glucose and heading down here towards pyruvate. And for gluconeogenesis, we are doing the opposite. So we're taking the pyruvate and building our way back up to glucose. Our pathway looked like this. So on the left we have glycolysis, so starting with glucose and heading down to pyruvate. And on the right we have this gluconeogenesis. The enzymes listed in blue are exact same for those same steps in the glycolysis. The enzymes listed in pink are those regulatory enzymes. And so, for example, here, glucose 6-phosphatase is the one we see in gluconeogenesis, takes us from glucose 6-phosphate to glucose. But over here in glycolysis, we had hexokinase going the opposite direction. So the regulatory enzymes work in only one direction. In this video, we're really going to focus in on just the substrates, products, and enzymes of these regulatory steps. So we're going to look at these three regulatory steps. Notice that we are starting up here with glucose and ending up in pyruvate. So along the left, as we go down this direction, we have glycolysis. On the right, in the blue, we would be going this direction for gluconeogenesis. Glycolysis occurs in all cells, but gluconeogenesis occurs mainly in the liver, to some degree in the kidneys, and to a very small extent they're discovering it occurs in the brain and other organs, but really mainly the liver, so we're going to focus on that. This glucose 6-phosphatase is only found in the liver, which makes a lot of sense because the role of the liver is to keep our blood glucose level over a 24-hour cycle, right, of eating and fasting. And so this helps keep our blood glucose levels fairly constant. If you remember, in glycolysis, hexokinase was regulatory, but not the key regulatory step. And the same is going to be true for the glucose 6-phosphatase. It's regulatory, but it's not the main regulatory step. If you remember, what really affected the hexokinase rate was the concentration of this glucose 6-phosphate. Right? So, as glycolysis steps further down here, slow down, glucose 6-phosphate, the product of that first step of glycolysis, would start to build up. And if it's building up, that indicates the rest of this is slowed down. We must not have high energy needs. And so this one would serve as a negative effector on the hexokinase. If we started seeing glucose 6-phosphate building up, it would slow down glycolysis. On the other hand, as that starts building up, it is going to positively affect our glucose 6-phosphatase. It is essentially a sign that, hey, the energy needs of the cell are probably met, so let's use this glucose 6-phosphate to build up our glucose. That it's okay to do that. All right, so that's that first step in glycolysis, last step in gluconeogenesis. And now, though, we really want to look at the steps that are more of our key regulatory steps. So as we talk about this, the big idea okay, is that the balance between glycolysis and gluconeogenesis in the liver is sensitive to that blood glucose concentration. So the blood glucose concentration is going to help determine which of these two, glycolysis or gluconeogenesis, should be moving forward. As we look at this, so the liver is trying to balance between glycolysis and gluconeogenesis to keep blood glucose concentration constant. Okay, we already looked at this top step, glucose 6 to glucose 6 phosphate. And so now what we're going to look at is the allosteric regulators of these other regulatory steps. All right, so focusing in on the pieces highlighted in green, big picture. If we have lots of glucose, need lots of ATP, we're going to expect glycolysis over here on the left to run fastest. On the other hand, if we need glucose, glucose is scarce, we're going to expect gluconeogenesis to run fastest. So keeping this big picture in mind is going to help us think about why certain allosteric effectors are either positive or negative effectors for each step. Okay, so we're going to focus in on this step in the green box. So this was the fructose 6-phosphate to fructose 1,6-bisphosphate. This was our PFK, key regulatory enzyme in glycolysis. Okay, and there are a lot of different effectors for this step on both sides, glycolysis and gluconeogenesis. And so we are going to start right here looking at these ones that I have boxed with the orange box, just to focus us in. All right, so picture this. In glycolysis, if our cell needs energy, or just in general, if our cell needs energy, let's write that, if cell needs energy, we'd expect to see our concentration of AMP increase. Okay, and hence, because of that, we see that AMP is a positive effector of PFK. It's going to help glucose move forward. Okay. On the other hand, if we have plenty of energy, okay, the energy needs of our cell are met, we're going to expect that we're going to have higher concentrations of ATP. Okay, and hence, if we start seeing lots of ATP, that's going to be a negative effector of PFK. It's going to say, hey, wait, we got enough energy. Let's stop this glycolysis. Okay, so remember, these are allosteric effectors, so the AMP, the ATP would bond to some regulatory site on our PFK and slow it down. Now, on the other side, it turns out that the one that binds to this enzyme, fructose 1,6-bisphosphatase, so phosphatase is removing a phosphate, and the one that really bonds here is the AMP. And so remember, if we see high AMP levels, just like over there on the glycolysis side, if we have a high concentration of AMP, it likely means we need energy. Okay, well, if we need to be making energy, we're going to need glycolysis running, right? We're going to need to be going this direction, not this direction. And so high AMP will slow down that phosphatase. We want to be moving towards the fructose 1,6-bisphosphate, not away from it. Okay, so that's kind of based on the energy indicators. Let's think about that H plus concentration. So as we think about this, a higher H plus concentration would mean a lower pH. So remember, those are inverse. Okay, so we're talking about pH going down. So when you are actively respiring, oxygen is low, glycolysis is running quickly, and your body is making lactic acid. Okay, on top of that, protons are a byproduct of respiration, the oxidative phosphorylation. So as all of this happens, we're making lactic acid, we're making these protons, and if the pH gets too low, it can actually damage the enzymes. So we want to prevent that. So this is essentially a negative effector of PFK. It'll slow glycolysis, and it's essentially a safety valve. It's going to slow glycolysis until that lactic acid can be moved out of the cell, until H plus can be moved out of the cell by various pathways to keep the enzymes from being damaged. All right, onward. Let's talk about citrate. So if we come over here in our lactic or in our metabolic cycle, we've got glucose, we've got pyruvate, okay, and then let's look, citrate is down here, okay. So citrate is a part of our citric acid cycle, and if we have made it to citrate, so we've gone all the way through glycolysis down to pyruvate, and now we've made it over here to citrate, and there's different pathways. That's an indication that we've been able to produce plenty of energy, we've got plenty of carbon for our biosynthetic needs, right? Because we've already taken that pyruvate and started making the citrate. So when citrate gets high, it's an indication that, hey, we don't need to keep making this stuff. We don't need to keep moving towards pyruvate. So it'll be a negative effector of PFK. All right. But when we have a high concentration of citrate, it says, you know what? We've got plenty of carbon for our biosynthetic needs. We've got plenty of energy. Let's send part of this back and start building up our glycogen stores, our glucose, right? So it's okay, so it's a positive effector, it's okay to push it back along this biosynthetic route where we're moving back up to glucose. Our energy needs are met, we got plenty of our carbon source. All right, that leaves just our fructose 2,6-phosphate, which we talked about in glycolysis. And remember that fructose 6-phosphate was a positive effector of PFK. It sped up, whoops, that's not what I wanted, PFK. It sped up our glycolysis, and as you might predict, it's going to be a negative effector, because when we want glycolysis to speed up, we're going to want gluconeogenesis to slow down. Let's review, okay? Glycolysis regulation of PFK by fructose 2,6-bisphosphate. So remember, this is not our actual substrate for glycolysis. That was the fructose 1,6-bisphosphate. But we had this side reaction. As fructose 6-phosphate builds up, we could have this side reaction that makes some of this fructose 2,6-bisphosphate. And that served as a positive effector on PFK. It sped up glycolysis. All right, so let's go back now and look at how it does that. Okay, bunch of words first. We're going to look at it and look at the enzyme. So in the liver, rates of glycolysis and gluconeogenesis are adjusted to maintain blood glucose levels. The key regulatory regulator of glucose metabolism in the liver is this fructose 2,6-bisphosphate. Okay, it stimulates the PFK and slows down the fructose 1,6-bisphosphatase. Speeds up glycolysis, slows down gluconeogenesis. All right, so careful here. The kinase that synthesizes the fructose 2,6-bisphosphate, so now this is a separate enzyme, not really in our glycolysis chain. It's for the side reaction. So for this side reaction, we have this going on, and this one is a bifunctional enzyme. All right, so we have this bifunctional enzyme happening, or that we use for this. All right, if you remember this from glycolysis,